Alright, Chapter 4, we're looking at organic chemistry, and this deals entirely with the element carbon, which is element 6 on the periodic table. Recall that the first orbital in atoms contain 2 electrons, and this image shows us the remaining 4 in the second valence shell. And then recall the second valence shell can hold 8 electrons, so that means that carbon needs 4 more electrons to be a happy atom. And this gives carbon the ability to form 4 different bonds, and we'll talk about the significance of that coming up here. Organic chemistry originally came from vitalism. This is the belief that organic compounds could arise only in living systems. However, this belief vanished as scientists learned how to synthesize organic molecules from pure elements in the lab setting. And as scientists moved away from vitalism, they started to adopt the belief of mechanism, which is the idea that physical and chemical laws control natural phenomena. Going back to those 4 unpaired electrons that carbon has, we can see that it has the ability to take on a lot of different shapes as it uses those unpaired electrons for bonding, and as mentioned, covalent, which is the solid bars that we see coming off the carbon atoms here. We have straight chains, branched chains where they branch out, straight chains sometimes involving double bonds, and different types of rings that we see going on here. Now the carbon skeleton or hydrocarbons, all these unnamed atoms that would be on this bond here and this bond here would be hydrogen. And hydrocarbons specifically contain only the elements carbon and hydrogen. And these are the dominant structure in most living things. They form critical fats, as we will see as we progress through this course. Some of these fats can be used for energy, although it is not our primary source of energy. Typically those are carbohydrates. But fats can pack on an extraordinary amount of energy that can be used, 9 calories per gram actually. Here we see some examples of different layouts that hydrocarbons can take with the hydrogens plugged in now. And if you look at these hydrocarbons here, they all have something in common. Yes, they both contain carbon, they both contain the hydrogen, but all 3 of these have 5 carbons, and all 3 of these have 12 hydrogens as well. So carbon, once again, with the 4 unpaired electrons, it can form bonds in different shapes and in different ways. Instead of having a straight chain of the carbons, we can have a branched one up here. So they can bond in different ways. Whenever we have things like this where the numbers of atoms 5 and 5 and 12 and 12 are the same, those are called structural isomers. They're the same elements, same number of those elements, just arranged and bonded together differently. Another type of isomer are the cis and trans isomers. As you can see, this is a fairly simple one. Cis-trans, they both involve a double bond between the carbons, which means that each carbon will have 2 more bonds to form, 1, 2, 1, 2. Now cis and trans, the difference between these two, in this case they're using chlorine for the extra item here. Chlorine are on both sides, or on the same side over here, excuse me, and here they're on opposite sides, hence the cis and the trans. What you should know is they are similar, they involve the same atoms and compounds and quantities, and another thing to also keep in mind is these can be longer chains of different things, not necessarily just one atom here and here. It's just, oops, these have to be the same as these in order to truly be the isomer form. And the third type of isomers are called enantiomers. These ones are essentially mirror copies of one another, same atoms, same quantities, same groups, COOH. They're just a mirror copy of one another. Now what we need to know about these isomers is, despite having the same numbers, same groupings, same here with the cis trans, and same here with the structural isomers, is that these different arrangements create entirely unique properties for each one of these. They do not behave in the same fashions as their relatives, so to speak. They behave uniquely. Now moving into the functional groups associated with these carbon skeletons that we spoke about previously. This one happens to be the linear format. The functional groups are things that may attach to the carbon skeleton. The first one we're going to talk about is the hydroxyl group, which is OH. Don't confuse this with the HO that's associated with pH. They're different functional groups. Although this could arrange itself as HO, it doesn't necessarily have to be OH. Typically it's OH though. Hydroxyl groups are alcohols. They all end in OL. Ethanol, methanol, a couple of different examples. Hydroxyl groups are polar molecules, and that results from the difference in electronegativity between carbon and oxygen here. So the electron from carbon is going to be pulled towards the more electronegative oxygen atom here, causing a polar molecule. Recall that polar molecules will be able to form hydrogen bonds between other molecules. And once again, hydrogen bonds, we see a hydrogen right here. The next functional group are the carbonyls. There's a couple different varieties, ketones and aldehydes. Now, different things are highlighted in red here, but we're looking at what's the same, and that's the carbon and oxygen with the double covalent bond between them. The difference between ketones and aldehydes is location of the double bond between the carbon and the oxygen. In ketones, we can see that it's kind of embedded in the middle of the carbon skeleton, and the aldehydes, it's at the end of it, and we have a hydrogen at the end on both of them. So really, we're just kind of looking at the double bond between the carbon and the oxygen, so CO. Also keep in mind with the carbonyls that structure and different structures lead to different properties and different functions. So even though these are both carbonyl groups, the location of the double bond CO group will create different properties between the ketones and aldehydes, so they do behave differently. Next we have the carboxyl groups. COOH is typically how it's seen in textbooks. It involves a double bond between one of the oxygens and a single bond between the OH portion of it, and the last one's tied into the carbon skeleton. These are also known as carboxylic acids or organic acids. An example would be acetic acid, which is found in vinegar. These act as an acid because in solution they can donate this hydrogen proton, which would increase the acidity of the solution. Next we have the amino group, which is, once again, we have our carbon skeleton, an NH2 group. This one happens to be glycine, and as we can see, it has a carboxyl group on this end of the skeleton, making this more than just an amino group. This is an amino acid. Once again, this hydrogen can be donated to increase the pH. I guess I should say lower it, make it more acidic. However, on this end over here, the amino group, the NH2, this can actually act as a base and accept a third hydrogen proton, making this end become NH3. So this would be an amino acid where it has an amino group and the carboxyl group over here that can act as an acid. The next functional group is sulfhydryl, which is an SH group containing sulfur. Once again, we're looking at another amino acid, amino group, acid group. This one happens to be cysteine. So, next are the phosphate groups. These contain a phosphorus bonded to 4 oxygen atoms, 2 of which have a negative charge, and one that is bonded to the carbon skeleton. They're written out as OPO3 2-. Glycerol phosphate here provides the backbone for the phospholipids of the cell membrane. We'll be looking at that in Unit 3. The negatively charged oxygens create a negatively charged molecule, so this is negatively charged as a whole. The last functional group we're going to be looking at is the methyl group, which is written out as CH3. We're only looking in this picture here at this one right here. This is the only one that has a methyl group. This is 5-methylcytidine. The methyl group, it can be attached to another carbon in the skeleton ring here, or it could be attached to another element or group. It doesn't have to be attached to a carbon. The addition of a methyl group to DNA alters gene expression, and male and female sex hormones have these methyl groups, which does ultimately affect their shape and their function. One thing before we do move on worth noting here is that when we're dealing with carbon and carbon ring structures, very commonly, like right here, there's an understood carbon right here. A lot of times the carbons just aren't written out, so this one actually has a carbon right here and a carbon right here, and I'm relatively surprised that they even included this carbon right here. This does not need to be written there. You can just put an elbow in. That's the only element that I've known about where they'll use an elbow instead of actually putting the C there. Just keep that in mind as we move forward and start looking at molecules of glucose. And then the last thing we'll be discussing in Chapter 4 is ATP, ADP. Not so much the energy part of it. We'll be getting that in Chapter 8. But we noticed something that we were just speaking about previously, and that's phosphate and phosphate groups. This one actually has 3 phosphate groups, as we can see right here. And when we release energy, we remove one of these phosphate groups from the ATP molecule. When this phosphate group is removed, we form an ion, HOPO3 2-. ATP can release that ion, and when that's done, we're left with 2 phosphate groups, ADP. ADP can store that phosphate ion. And once again, just the last thing I wanted to reiterate, each of these elbows, where nothing is written, is an understood carbon. So in this sugar right here, ribose, we have 1, 2, 3, 4 carbons in it that aren't even written. Don't overlook those. There's no carbon right here because oxygen is right there, so that's an understood oxygen. This concludes Chapter 4 and Unit 1. Make sure you review your materials because our first test will be coming up shortly. If you have any questions, please bring those to class, and we can nail them out together.